Okay. Good afternoon, everyone. I, Devashri from the SciBug team,
Good
would
afternoon.
like to welcome you all for today's session. Today's webinar on bioinformatics tools for nucleotide to protein translation and overview and access links is brought to you by SciBug. Before starting the session, it's a humble request to all the participants to please keep your audio and video settings off in order to avoid disturbances during the session. Now I would like to welcome Dr. Bharti. Dr. Bharti Wadekar is a renowned microbiologist with a PhD and over 9 years of experience. She serves as a faculty member at Thakur College of Science and Commerce. Her research focuses on probiotic properties of marine microorganisms and bacteriocin production. She has authored multiple research papers and books in reputable journals, receiving multiple awards such as Best Paper Presentation and Indian Woman Talent Award. She has also received honored with the Global Youth Icon Award for her impact on the scientific community. Recent publications include studies on Enterococcus species isolation, phenol degrading microorganisms, and biofertilizer production from rhizobium species. She has 3 patents granted for novel bacteriocin composition and 2 patents applications published. Dr. Wadekar actively participates in educational policy discussion, publishing insights on India's national education policy and its implications for languages, arts, and professional education. As a dedicated scientist and educator, Dr. Wadekar bridges research and practical applications. Her contributions inspire students and fellow researchers, making her a valuable asset to the scientific community. Through ongoing research and advocacy for educational reforms, she promotes a deeper understanding of microbiology's relevance in addressing contemporary challenges. Dr. Wadekar's work exemplifies her dedication to advancing microbiological research, education, and environmental sustainability, solidifying her position as a leading expert in her field. I would like to thank Dr. Wadekar for accepting my invitation and taking the time out of her busy schedule to join with us for today's webinar. Now, I would request Dr. Wadekar to take over. Over to you, ma'am.
Right. Ma'am, please
Yes,
share the slide.
yes, ma'am.
Okay. Shall I start?
Yes, ma'am.
Okay. Good afternoon, everyone. Today, I will discuss on the topic bioinformatic tools for the nucleotide to protein translation and overview and access link. See, this is very easiest topic. How to find the ORF, that is open reading frame, and this ORF is used to for the translation process. Okay. So, bioinformatics has revolutionized the way we analyze the biological data, particularly in the field of nucleotide to protein translation. So, these various tools exist to facilitate this process, helping the researchers, scientists predict the protein sequences from DNA or RNA. These identify coding regions and study the protein sequence. For that purpose, use the some essentials tools include the RF finder, first one, that is used to find the start codon to stop codon. Okay. Now next one, XPASI translate tool, which is used to find the coding sequence. Then BLAST as all of you know about the basic log alignment search tool, which is widely used for the comparing of nucleotide or the protein sequences against the large database. Now, next one is the gene mark. Gene mark is one of the another tool for predicting protein coding gene in DNA sequence, particularly useful for the genomes that are all not well annotated. So use the gene link. All these tools streamline the translation of nucleotide sequence to the protein. Next slide. So purpose of this seminar is used to provide the overview of the open reading frame and its role to understand gene structure and the function. So objectives to give an overview of ORF and its significance. These ORF are the significant because they provide the insight into the coding region of the genes, which are essential of the protein synthesis. Understanding the ORF, which can help in predicting the functions of genes and their encoded protein. The next objective to learn how ORF Finder searches for open reading frames in a DNA sequence using understand or alternative genetic codes. This ORF Finder is very powerful tool that helps researcher identify all the possible ORFs within a given DNA sequence. ORF Finder is used to find the possible open reading frames within the given DNA sequence, which is enabling efficient annotation of the gene, which will explore how use this tool or how work this tool, and including ability to use the different genetic codes to account for various variations in the translation across the species. Now, third objective is to identify all the possible open reading frames in a sequence. So, this tool is used to find the total 6 possible reading frames and identify the potential ORFs. Now, last objective to study how to perform the blast search in a particular ORF. That is, blast search is used to identify this protein sequence found in a database that identify the homologies, possible functions, then evolutionary relationship within the known sequences. Known sequences means which DNA available. DNA sequence, which is the any DNA sequence available with you. Okay. Now next slide, ma'am. What is ORF? ORF Finder is a bioinformatic tool used to identify the open reading frames within a given DNA sequence or available sequence. Then ORF are the scratches of nucleotide that have the potential to be translated into the proteins, which is starting with the start codon, usually AUG, and ending with the stop codon, usually UAA, UAG, or UGA. So main concept of ORF finder, first one is the ORF, already told about. It's a continuous stretch of the nucleotide that begins with the start codon and end with the stop codon. Then DNA strand and the frames. We know about the DNA strand. Everyone know about. So DNA strand is a double stranded that have the complementary strand and the template strand, whatever. Okay. Then ORF can be found on both the forward as well as the reverse strand of the DNA sequence. Then start and stop codon. Everyone know about the start and stop codon. So this ORF finder tool looks for the start codon, usually AUG in RNA or ATG in DNA, to initiate the translation process. Why necessary to create the start and stop codon? To initiate the translation process. Then it also scan for stop codons to terminate the translation. First one is the to scan or to predict or to detect start codon, and next one is the to detect the stop codon. This is the one of the most part of ORF finder. Next slide, madam. Now, how to find ORF? So, to locate an open reading frame, let us consider the hypothetical sequence, nucleotide sequence. Hypothetical, assume it. This is the hypothetical sequence. Okay. Then, in step one, divide this sequence into the 6 different reading frames. 6 different reading frames. So this sequence can be read in 3 possible frames on the forward direction, that is +1, +2 and +3, and 3 on the reverse complement strand, that is -1, -2 and -3. There are the 6 reading frames. Okay. Now next, identify the first reading frame, that is +1. So to find the first reading frame, we start from first base and uh, into the sequence, group the sequence into the codon, that is the 3 sets of nucleotide. 3 sets of nucleotide, that is the CGC, that is the 3 set of nucleotide, that is one codon. Like that, we have to prepare the 3 sets of codon, 3 sets of nucleotide of given हाइपोथेटिकल न्यूक्लियोटाइड. क्या करना है, सारे तीन तीन कोडॉन का एक सेट बनाना है, दैट इज द रीडिंग फर्स्ट रीडिंग फ्रेम +1. आपके पास जो भी सीक्वेंस मिला है डीएनए सीक्वेंस, फर्स्ट वी क्रिएट द sets of 3 nucleotide. Okay, in this frame we scan for the start codon, then we try to scan for the start codon as well as the stop codon. See, listen, this is the hypothetical sequence, example of nucleotide sequence. First part, divide the sequence into the 6 different reading frames. Okay. Now, next one is identify the first reading frame, that is the +1. I already told about what there are the 6 reading frames. So, for in a given sequence, we divide first reading frame, that is +1. We already identify +1 frame. Okay. Now next slide. So now next slide. In the next slide, we find the next second reading frame is formed by skipping the first nucleotide of the sequence and dividing the remaining sequence into the codon or the again set of the 3 codons. See here, the C left out. First nucleotide we have to skip out. Okay, skip out and then prepare again the 3 set of the nucleotide. Prepare codon of the given sequence. Similarly, the third reading frame, that is the frame +3. In that, we have to skip the 2 nucleotide, that is CG. We have to skip that one. Okay, and then again prepare the set of 3 nucleotide. Got it? What is frame 1? Frame +1, frame +2, frame +3. We have this given sequence. In sequence, frame +1 create the simple set of the 3 nucleotide. Then next frame +2, skip 1 or skip first nucleotide and then again create the set of the 3 nucleotide. Then frame +3, skip first 2 nucleotide and create the set of the 3 nucleotide. Okay, each of these frames many contain the potential open reading frames, that is ORF. ORF is one of the sequence which is start from the initiation codon and end with the stop codon, which can be identified scanning for the start and the stop codon, and it is very important step in finding all the possible protein coding regions. Next one. See, we work on the forward strain, forward frame +1, +2, +2, +3. Now, Other three reading frames are found using the reverse complement strain. First sequence, that is complement, already given, but we prepare the reverse complement sequence. Reverse complement sequence, it is started from the last nucleotide, that is the A, T, G, G. See here, in the first sequence, A is the last nucleotide. So in a reverse complement, it comes in a first. So A, T, G, G, C, this type of the reverse complement sequence we have to prepare, and after that we will get the next three sequence open reading frames, that is the frame -1, -2, -3, and same procedure for the frame -1. Start the reading from the first nucleotide, that is, and create the set of the three nucleotide ATG, GCT, AGC, like that. Then frame -2, skip the first nucleotide and then group in the set of the three. Then frame -3, again same, skip the set of the first two nucleotide and then group in a set of the three. Like that we got the, there are 6 reading frames: frame +1, frame +2, frame +3, and frame -1, -2, -3. -1, -2, -3 is coming from the reverse complement. Okay, next slide.
Hello,
Yes, yes sir.
ma'am. In the last slide, in frame -1, -2, -3, we have to nullify the base from the like latter portion from the finish. We have to nullify the base. Just like in case of plus, we have neglected the base from the starting, like A, then I have described for first base, then second base, then couple of base at +3. Just like that, in case of -1, -2, -3, it will be from the finish portion or anything else. I could not actually follow in the last slide.
See, listen here. In a first three frames, we use the given sequence, right? Given sequence, that is, it started from the G to completed up to the A. Then we create the set of the three nucleotide, right? That is the codon. Okay. Then after
Okay.
that three nucleotide, we prepare the frame +1. Frame +1, keep as it is, no skipping of the any base pair.
Okay.
Okay. Then frame +2, skip first nucleotide, that is C.
Okay.
In a given के बारे में बोल रही हूँ, not a reverse. Okay. Then next, in a frame +3, skip first two nucleotide and again prepare the set of the three nucleotide. Got it, my point?
Okay. Okay.
Okay. Now for the reverse, we have the given sequence. Then this sequence we turn into the reverse format. That is, first given sequence start from the G and completed to the T. Okay, sorry, A. Okay. Now in a reverse sequence, the sequence is start from the A and completed to the G. Read out. A, T, G,
Okay.
G, ये पूरा रिवर्स हो गया, right? And then after that we got the minus reading frame. जो रिवर्स से हम लोग रीडिंग फ्रेम प्रिपेयर करेंगे, that called as a frame -1, -2, -3, and same procedure as it is frame +1, +2, +3. Got it?
Okay, okay. So the procedure is same, but we have to take the complement of whole series.
Yes
We
sir,
have
yes,
taken the complement. Okay, fine. Thank you.
yes. Okay. Now next, okay, minus frame. I already told minus frame. For the reverse complement sequence, we can identify the three reverse reading frames. So frame -1, starting from the first nucleotide of the reverse complement is the ADG. We divide the sequence into the codon, three codons, and we got the like that of the sequence GCT, AGC, and end up to the last three sequence code is a TAG and remaining is CG. Okay. Now next frame minus 2, we again skip the first nucleotide and then divide into the sequence into the set of the three. A, skip out and again prepare the set of the three. Then frame minus 3, we again skip the first two nucleotides and then again prepare the sequence into the set of the three. That is AT, skip out, then GGC, TAG, CGA up to the GCG. So these all are 6 frames, reverse and a forward. First forward plus frame plus 1 to plus 3 and reverse sequence frame minus 1 to minus 3. By analyzing all 6 frames, forward strand and the 3 on the reverse strands, we can identify all the possible open reading frames, maximizing the chances of locating potential coding regions. Okay, got it? Now next slide. Okay, we got 6 reading frames. Now we start the specific open reading frames. That's, let's identify the start codon and the stop codon from that the different reading frames. So frame plus 1, there is no start codon, but stop codon is available. That is TAG appears near the end of the sequence TGC and GTT. Okay. Now in a frame plus 2, we see the start codon in the middle. Got it? That is ATG available in the middle of the CTC, ATG, GAT. Then again we find a stop codon and got it in the TAG appear near the end of CGC, TAG, CCAT. See here, in a frame 1, only stop codon we got, no start codon. In a frame 2, both codons available, start codon and the stop codon. And in frame 3, again we find start codon ATG in the middle, but no availability of the stop codon. So I already marked this start and the stop codon, so we can begin to identify the potential open reading frames which may represent the protein coding regions in the sequence. We got in a frame plus 2 start codon and a stop codon. Okay, means about this given or finding 3 frames, we got only 1 frame has start codon and a stop codon. Next slide. Now let's identify again start codon and the stop codon in a reverse frame. In a frame 1, we see the start codon, that is ATG at the beginning, and stop codon TGA in the middle. Okay. Now same in frame minus 2, there is presence of the start codon ATG in the middle and stop codon immediately after. We find immediately after the stop codon, that is TGA. Okay. Now next in a frame minus 3, in a frame minus 3, the start codon appears towards middle the TCCATG, and the stop codon also available and present near the end of the CCAGCC. In 3 frames, we find start codon, stop codon, both are available. So by identifying this start and stop codon, again we find the potential ORFs in the reverse complement of the sequence, which is used to analyze all the possible coding regions. Next slide. See. Now let's identify the open reading frames in the sequence provided. Firstly, we got the frames. Then we find the start codon and the stop codon, total sequence. Then we separate out this ORF, open reading frame, means the sequence is start with the initiation codon ATG and end with the stop codon TAG. So out of that 6, we got 3 open reading frame. मीन्स आपके पास जो भी सीक्वेंस है उसका पूरा ओपन रीडिंग पहले फ्रेम बनाना, फ्रेम बनाने के बाद जिस जिस फ्रेम में आपको इनिशिएशन कोडॉन और स्टार्ट कोडॉन एंड स्टॉप कोडॉन का सीक्वेंस मिलता है, दैट कॉल्ड एज अ ओपन रीडिंग फ्रेम. आउट ऑफ दैट 6 we got only 3 open reading frames. Which one is the frame -2, frame -1, frame -3, which are started with the start codon ATG and stop with the TAA or TAG. So identifying this ORFs is important for predicting the potential protein coding regions, providing insights into the gene functions and the expression within the genetic material. Next slide. Now we got the codons or the start codon and the stop codon. Out of that, after that, we start the translation procedure. Translation procedure, we give the amino acid sequence. So frame +2 ATG, that is methionine, and the NTAG, that is the stop codon. So we got the amino acid sequence. This sequence of frame 2 translated into the different amino acid sequence. Same frame 1, we got again amino acid sequence. Then frame -3, we got the again same amino acid sequence. This amino acid sequence we predicted, we find or we assume we got the one of the protein, right? By analyzing this ORF sequence, we can predict the possible amino acid which is produced during the translation process. So this prediction of the correct ORF from the newly sequenced gene is a critical step in understanding the gene functions, which is used for designing the primer in experiment PCR, sequencing, then other further research work also used in application in a molecular biology. So these 3 frames used to prepare translation procedure, that is amino acid sequence, and we got the one of the protein. Now next PPT. This is about ORF finder program, that is bioinformatics tools. Up to I told about the theory of ORF. Now let's open the browser and navigate to the home page of the ORF finder. You can access it by going to the NCBI website, or as well they have own page ORF finder. Next slide. Firstly, I will tell about theoretically, then we will do the practical on a website. Okay. So in this slide, on the ORF finder main page, you will find the text field where you can enter the accession number or give one sequence or FASTA format of the DNA sequence. Okay, so FASTA format is generally one of the simplest sequence format. It begins with the greater than symbol. Whenever we got any microorganism, then we done the what? Sequencing process. Then we got the FASTA format, and FASTA format is always start with the greater than symbol, and in additional comments which is followed by the actual nucleotide sequence itself. This page or a finder and this small box shows small box. In a small box, you or we input the one of the sequence of any microorganisms or FASTA format of microorganism or accession number of microorganism. Next slide. Next slide. Then here we can see a text field already told about enter the accession number or the text to enter the query sequence in the FASTA format and run or click on the button to run the ORF finder. Next slide. See, we put the FASTA format or the FASTA sequence of given microorganisms. See like that. Next slide. Click on the genetic courses. Genetic codes means which is related to the human microorganisms, archaeas, bacteria, protozoa, etc. The next slide. These are the standard vertebrate, yeast, mitochondria, all are the standards. Which type of sequence you put in a box, that you have to click anyone, that is आपने बैक्टीरिया का सीक्वेंस डाला है तो क्लिक ऑन द बैक्टीरियल कोड, माइटोकॉन्ड्रिया का सीक्वेंस पुट किया है तो देन क्लिक ऑन द ईस्ट माइटोकॉन्ड्रिया, वट एवर रिलेटेड टू द माइटोकॉन्ड्रिया. ओके, नाउ नेक्स्ट स्लाइड. See here the example standard code which is UGC code for the methionine, and different codes like vertebrates mitochondrial codes, yeast mitochondrial code, AUA code for the methionine. Then we press the ORF finder button, then get the result. The result shows all the possible 6 reading frames which is present in an entire sequence query, that is the FASTA format. One can see the ORF is listed according their size and the graphical representation of the sequence. If you put the sequence in the box, then click on the genetic code and then click on the ORF button. We will get the graphical representation of the sequence. Next slide. Like that. This is the name of the any organism, and these are the ORF sequence. This see +3, -2, -2, -2. These are the +1, +1. These are the number of sequences. Next, numbers of ORF readings. Okay. Next, see, whenever we click on any button, that is +3 or the green button, then this purple color shown here in a graphical representation. Click on green color button which is represent this ORF sequence in the purple color. Okay. Now next slide. Next slide. After that, simply only +3 we click represent on the graphical represent on the purple color shown on the graphical representation. So after that we click on the accept button. So accept button shows this type of the translation codon, right? Number of amino acid sequences, and in below we got the some simple name GF, valine, isoleucine, then glycine, alanine code, that is amino acid code. Okay, now next slide. Same, next slide. Okay, so you can also perform the blast search for the particular ORF that we selected and select the appropriate program and the database, then click on the blast button. क्या किया है बाद में, you click on the blast button. After that we got the name of that bacteria or the any specific number of that protein sequence. Next, see, BLAST is a pairwise sequence alignment tool which compares nucleotide or the protein sequence, and there are the types of the BLAST and which is used for the different program and use the different algorithms. So nucleotide BLAST search the nucleotide databases using the nucleotide queries, and their algorithms we mostly use the BLASTion, megablast, discontinuous megablast. Like that, these are the different types of the blast. Next, after that, click on the blast. We got the report. So in a report, click on a view report and download the report. Next. So above is the following is the blast resulting page. You can see the alignment score and the description here. In the next slide, we got the alignment score and a description. Then after submitting the query sequence for the sequence similarity, the result page will appear along with the information like query ID, description, molecular type, length of the sequence, database name, program, blast program, etc. It shows the putative conserved domain that have been detected while undergoing sequence similarity search. Next slide. See, again, we got the graphical representation of the protein sequence. Firstly, we put only the nucleotide sequence. After that, we find the ORF. After that, we click on the BLASTA button, and BLASTA shows give you the protein sequence, graphical representation of the protein sequence. Next, so this alignment is proceeded by the sequence identities along with the definition length of the match sequence, followed by the score E. coli. All are related to the algorithms. The line also contain the information about the identical residues in the alignment, their positivity, number of gaps which are used in the alignment. Next slide. All experiment you have to do, and on a NCBI database there are the different links, ncbi.ni nim nih nlm nih.gov, orf finder, and then project or govs. Madam, please slide. Thank you, thank you. Now we start. Please stop the sharing. Now we start the website work. How to get ORF slide, ORF finder. Okay. Is it visible? See, directly go to the Google Chrome. Then we type the ORF finder and home page. Click on the home page. Okay. Then this one is the, now we put the one of the FASTA format of any nucleotide, or this is the example. If you have any one nucleotide, please tell me, share me with me. Otherwise, I will click on the one of the example. Can I click on the example or use one of the any nucleotide sequence? Anyone tell me? I have used the one of the bacterium sequence.
Use anyone, ma'am.
Use anyone? Okay, thank you. See. Only sequence, faster format.
Ma'am, which one you have used just? You have pasted the which faster format?
It is related to the Lactobacillus acidophilus.
Okay, so that is like given as an example, or you have given from your side?
My side, from my side.
Okay, okay, okay.
Is it example of already given here? That one, that two example already given here. You have to click here. You got the sequence or their number. That is the NC001604. Here number, click out, and then already put up the minimal ORF length. See, now this is my given sequence. ORF minimal number you can put up to the 600, depend on the FASTA range. Okay, so I put the 150. Then genetic code. Genetic code, it's related to the bacterial, archaeal, and the plastoplastid. Okay, then ORF start codon, use ATG or alternative. Alternative is the another one. Which one? ATG है ना? Okay,
Yes,
fine.
ma'am.
Yes. So ATG only. Keep. Okay. Then submit directly. See what happened. ORF found only 6. Genetic code 11. start codon ATG. Okay, see here. ORF1. This is the marks ORF. Click here. So represent here. Now next ORF4. Click here. Represent here. ORF3. Click here. Represent here. Got it? Now see. I click on the ORF3. Okay. See here display. ORF display is related to first one is nucleotide sequence. This is our sequence that start codon is ATG and end codon is the TAG. That is, it is one of the open reading frame, name as a open reading frame 3, right? Next, display ORF on a nucleotide sequence, already done. Okay. Then protein sequence. This is the protein. Why? Because 3 sets of the nucleotide shows the one codon, amino acid. Okay. Then display ORF CDS translation. Now see, that is ATG methionine. Then this D, R, S, P, these are the number of amino acid. There are the 21 amino acid available here, right? Got it my point? Whenever you click on any ORF, you got the display, different display ORF as a nucleotide, display ORF as a protein sequence and as a CDS translation. Directly we got the translation, that is protein sequence. Now we click on the BLASTA. After that BLAST database, Swissport give the one of the, did you get? Stop. Now again I share.
Ma'am,
BLASTA.
here after, hello.
Yes, बोलिए. ये रहा आपका blast sequence. That is,
So like after blasting here, that will somehow similarity check from the UniProt database. Like that, it's work.
yes ma'am. Yes sir.
Okay.
See, these are the number of protein sequence shows the similarity of the different enzymes, proteins, binding proteins, which is available in the musculus, Homo sapiens, Pseudomonas, Candida. These are the different. Which one is the sequence? I will put, I have put Lactobacillus acidophilus. But after that ORF reading, we click on one of the ORF reading, that is -3. But -3 gives the one of the protein, gives the different protein sequences which is available in a different organisms, means shows the similarity. Got it? This is the last part, BLAST protein sequence, that is the ID. Who gave Swissport? Their again query ID, their description, and these are the graphical representation with the tree of the results. Whole thing. Got it? Is there any issue? यहाँ तक समझ में आया?
Yes, ma'am.
Okay, thank you. Now next one, we again try with their example. Click here. That is accession number. Okay. Next, you have to put one.
Your screen is not getting shared. Actually.
Okay. ORF finder. Share. Okay. See. I click here. So I put the accession number. Then their minimal length, keep 150. Related to the bacterial because name as I think salmonella. Then ORF start codon ATG or alternative. Now then submit. Whenever we use the ORF start codon ATG and alternate, then we got the number of ORF. How many here? 100. We got 100 ORF, genetic code 11 and start codon is ATG and alternative. Now click here 15. There is 15. Yeah. If you click here, represent here 15 15, that one. Okay. Then after 15, see here. CDS or 15 CDS, their location is start from the 9 already given. 9907 and the stop codon where present 10908 sequence. Okay. Now next display as a nucleotide sequence. That one start with ATG and end with triple T TAG. Okay. Now protein sequence already given. Then CDC translation. Then translation and after that blast. Searching. Yeah. See. Which sequence? Salmonella, right now. But which proteins got? First one is the ributherine, then peptidoglycan, and these same sequence, protein sequence available in a different microorganisms or different living beings. It's a very lengthy, I think. But some it nahi. Then there's scores, e-values, and accession numbers of that microorganisms. Complete. Okay. Anyone have issue? Please tell me. It's very easiest. You can open the homepage of the ORF finder, then play with the ORF finder. आपको फिर पता ही चल जाएगा क्या 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 और it's amazing. जो बायोइन्फॉर्मेटिक टूल्स रहते हैं very very amazing. Yes. Yes. Madam already. Madam will provide you PPT. Anyone?
If you have any questions, you may ask.
Ma'am, I have a question.
Yes, sir.
Ma'am, does this ORF finder tool works on virus genomes where there are various kinds of genome types available?
Yes, if you have a sequence of virus, you can try.
Like, ma'am, positive sense RNAs or negative sense RNAs, it can also
Yes.
transcribe them as well.
Yes, yes. Next.
Participants, if you have any questions, you may ask.
Ma'am, can it does the reverse process as well, like taking a protein sequence and getting the genetic sequence of it?
Means एक बार फिर से.
Ma'am, if there is a amino acid chain sequence we have, and then we use that amino acid chain sequence to get its genetic sequence, the DNA sequence from the protein sequence. Can it does that?
No, no.